Moving on now, we're going to discuss DNA recombination events, and we're mainly going to be focusing in on what's happening in bacteria with this. So this will be a new concept for us with genetics. All right, so typically when you think about genetic information being passed along from cell to cell, we think about reproduction. So we think about a mother cell or a parent cell dividing and creating two daughter cells, so one cell to two cells. And when that takes place, the DNA inside that original mama cell has to be duplicated or replicated so that each daughter cell has a faithfully reproduced copy of the chromosome or chromosomes if we're thinking about organisms that have more than one of those. All right, and that is what we typically think about. However, and that's from one generation to the next, parent to offspring. However, in bacteria, there are some methods by which DNA can be transferred from one cell to another within the same generation. And as we're going to see, this is one of the means by which drug resistance, we're hearing all the time now about antibiotic-resistant bacteria. So that's a trait. How is that trait being transferred out in the wild? Is it just through reproduction? No, as it turns out, in some cases, bacteria that are resistant to a particular type of drug can transfer the gene that is allowing them to be resistant over to a cell that wasn't resistant to it. And that's an example of what we call genetic recombination. Disinfectant resistant. The ability to use certain kinds of nutrients, production of toxins, as we'll see when we get into Chapter 11, the bacteria that make us sick, they very often do it because they produce toxins of various kinds. And so those are usually proteins that are damaging to our bodies. And since they're proteins, they're going to have genes that code for them. So there's a gene for that toxin, and it has been discovered that in some cases, cells that have the gene for that toxin can transfer it over to cells that did not. So this is a method for transferring beneficial traits to members of the species who don't already have that trait. And I know some of these things, and beneficial in terms of benefit to the bacterial organism, not to necessarily to human beings. All right, so now what I was just talking about where DNA gets transferred from a parent cell to the two daughter cells, that's also called vertical gene transfer because you're going from one generation to the next generation. And we're going to hear that term again because we're going to hear about vertical transmission of microorganisms where when we get into Chapter 11 and we talk about infectious disease in more detail, if a microorganism is transferred from a mother to a developing fetus in the womb, that's an example of vertical transmission from parent to offspring. Now, what we're going to be mainly focusing in on in this video clip is called horizontal gene transfer because it's occurring within the same generation. It's not parent cell to offspring cells. It's from one cell to another cell in the same generation. And another term for this can be recombination. Recombination also refers to DNA has moved out of one cell or one organism into a new cell or a new organism where it did not exist before. You are recombining the genes in new ways that they didn't occur in the past. That term recombination is also used when we start thinking about biotechnology and genetic engineering and genetically modifying organisms as well. A lot of times that's called recombinant DNA technology because we are artificially moving pieces of DNA from one organism into another one where they did not exist before. Okay, so horizontal gene transfer and recombination, we can kind of think about those as being meaning the same thing for our purposes in this class. Okay. Now, how does this happen in bacteria? There are several different methods by which some bacterial species can transfer DNA from one cell to another. And sometimes they do it actively, sometimes they do it passively, as we'll see. And we're going to go over a few examples just so you're familiar with them. The first one we're going to talk about is transformation, and unfortunately, with this chapter, there are a lot of words that sound the same. So I know we've had transcription and translation and now transformation. So you just have to be sure you're learning the terminology and learning how to use it properly. What we're seeing here, we've got an example bacterial cell. This is supposed to be its existing chromosome. A, B, C, and D are supposed to be 4 example genes on its chromosome. Over here we have a piece of DNA from a donor cell. So that cell has died, but its DNA is left over. DNA can be fairly stable. It doesn't break down all that quickly. So after a cell dies, its double helix DNA molecules may persist even after the rest of the cell has broken down and has been degraded. So we've got a little piece of DNA left over from some dead bacterial cell. And in some bacterial species, not all of them, and this is a very rare event, but sometimes it happens, this piece of DNA is taken up into a bacterial cell. And that's pretty remarkable when you think about it. DNA molecules are big for organic molecules. And we've talked about how, big stuff just does not move through a cell membrane. Even relatively small organic nutrients like amino acids and sugars need transport channels or carrier proteins to move them across the membrane. And so you don't have carrier proteins or pores that are big enough to let a big DNA molecule pass through, because the DNA molecule is way, 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 way bigger than a protein or a protein pore present in a cell membrane. So how that works, how do these bacteria take up these pieces of DNA? That's still not all that well understood. But occasionally it happens. And in some instances, all or part of this DNA will become incorporated into the DNA of the recipient bacterial cell. And so that's what they're showing you here. And this is called recombination. This is a recombination event because this DNA from the donor has integrated into the DNA in the recipient cell. So you've had some recombining going on. And now this recipient cell has a new version of gene A. So it used to have the uppercase version, now it has the lowercase version. And so this is now a genetically transformed cell. This is called transformation, because if this gene is coding for, in some way it's changing a trait or traits of this cell, that's where the transformation part of it comes in. Its phenotype has been transformed. Okay, how did we figure out that genetic transformation takes place? And interestingly enough, there was a microbiologist named Griffith who did a DNA transformation experiment with an organism called Streptococcus pneumoniae. And I'm sure you can figure out what respiratory disease this is a bacterial organism, pretty common cause of pneumonia. He did a really interesting experiment that actually predated our understanding of how DNA to RNA to protein works. Before all of that was worked out, he did a really interesting experiment. And so what he did is, so Streptococcus pneumoniae has different strains. There are strains that make capsules, and those are called encapsulated strains. So what they're showing you here in brown, those are a couple of coccus-shaped bacteria linked together. And this is a streptococcus, so in reality you would have probably longer chains than that. And the orange around it is supposed to represent a capsule, like we've talked about before. And then there are non-encapsulated strains of this organism as well. So they lack the gene or genes that allow them to make capsules. So they're depicted here. They don't have the little orange zone around them. So Griffith knew if he injected encapsulated Streptococcus pneumoniae into a poor little lab mouse, the mouse would die due to the infection. And he also knew if he took a sample out of that dead mouse and swabbed it onto an agar plate in the laboratory, colonies of Streptococcus pneumoniae would grow back out, And encapsulated bacteria, when they form colonies on agar plates, the colonies often look wet and slimy because the capsule attracts a lot of water. So he could tell that the colonies were coming from encapsulated Streptococcus pneumoniae. He also knew if he took non-encapsulated Streptococcus pneumoniae, injected those into a mouse, the mouse would remain healthy. If he took out a sample, swabbed it onto a plate, he would get some colonies of these non-encapsulated Streptococcus pneumoniae. So they were able to live in the mouse for a while and reproduce, but eventually the mouse's immune system took care of them and prevented the mouse from becoming sick. Okay. Now, he also knew, and this will seem kind of obvious, if he took encapsulated Streptococcus pneumoniae and heat killed them, injected those into a mouse, the mouse remained healthy. And if he took a sample out of the mouse, swabbed that onto an agar plate in the laboratory, he couldn't culture any Streptococcus pneumoniae back because the bacteria were dead before he injected those into the mouse. Okay, so that just seems like common sense. But the last thing he did, this was the really interesting thing he did. He injected a mouse with dead encapsulated Streptococcus pneumoniae and live non-encapsulated. So neither of those two things should kill the mouse, but when he injected them together into the mouse, the poor little mouse died. When he took a sample back out, swabbed it onto an agar plate, the colonies that grew back out contained encapsulated Streptococcus pneumoniae. And so that was a unexpected finding, because either one of these things by themselves would not have killed the mouse, and you would not have gotten encapsulated Streptococcus pneumoniae back. So this was his DNA transformation experiment, because the only explanation was that somehow this genetic characteristic, this genetic trait of being able to make capsules, was transferred from these dead bacteria to these live non-encapsulated bacteria, and they became encapsulated, and they were able to survive and reproduce and build up their numbers in the poor little mouse, and went on to kill the mouse. So this was a first example of genetic transformation, and we now know that the way this worked was that there were DNA fragments left over from the dead encapsulated bacteria that were taken up by some of the non-encapsulated, and they now expressed that trait of being able to build capsules, which helped protect those bacteria from the immune system of the mouse. All right, another technique that some bacteria have for transferring DNA is called bacterial conjugation, and we've talked a little bit about that before. So this is an image of a cell right here that is capable of doing bacterial conjugation. And it has sent out a structure called a sex pilus. We talked about fimbriae and pili in Chapter 3. So this is basically a protein tube built from pilin proteins. And it has connected that tube over here to this recipient cell. And some DNA of this cell will pass through that sex pilus or conjugation pilus and enter this cell. Okay, and that's another way that DNA can move from one cell to another in the same generation. This is called bacterial conjugation. The sex pilus is not always that long. Here's another image where you've got the mating bridge. There is the donor cell, the recipient cell, and there's a shorter sex pilus linking those two together. F plus and F minus here. F plus is referring to a cell that has the genes that allow it to build a sex pilus and do recombination. An F minus cell is one that can't build a sex pilus on its own. It's considered to be an F minus cell. All right, here's another depiction of this. So we've got a donor cell and a recipient cell, and the F plus cell. This is a plasmid, an extra piece of DNA in here. And that plasmid has the genes on it that allow a sex pilus to be built. So that's sometimes called the F factor, a plasmid with those genes on it. So this cell is able to send out a sex pilus, which is a protein tube, connects it to this one, and it's going to send this plasmid through that sex pilus over into this cell. So it's replicating that plasmid and sending a copy over into this recipient cell. And now notice, because this cell now has this plasmid, it becomes an F plus cell. It now has the genes on that plasmid that will allow it to build a sex pilus. And plasmids are pieces of DNA, so they can have multiple genes on them. So sometimes these F factors or F plasmids will also have genes that code for things like antibiotic resistance. And so as that plasmid gets transferred over to this recipient cell, now this recipient cell can become resistant to an antibiotic or a disinfectant, or maybe it can use a nutrient it wasn't able to use before. It just depends on what kinds of genes were transferred over. So that's called bacterial conjugation. That's another method of horizontal gene transfer. Sometimes these plasmids, these little extra pieces of DNA, when they have genes on them that allow bacteria to be resistant to things like drugs or disinfectants or antiseptics, those can be called resistance plasmids or R factors. That's another name for it. And that's somewhat older terminology, but you may see it be used from time to time. All right, another method of horizontal gene transfer is called transduction. So here's yet another T word that starts off with trans. So you got transcription, translation, transformation, transduction. So you just want to make sure you've got your vocabulary straight. So this is kind of an interesting horizontal gene transfer process because it uses bacteriophages. Bacteriophages are viruses that infect bacteria. So viruses, we've learned that they need a host in order to be able to reproduce. And even bacteria can become infected with viruses. Those viruses are called bacteriophages. All right, the bacteriophages that are going to do this transduction process, they have DNA genomes. Now, when we get into Chapter 5, we're going to see that not all viruses do, but the ones that perform transduction have DNA genomes. And they're viruses, so their genomes have to be, when they go into a host cell, their genomes have to be copied inside that host cell, and then they have to be packaged up into new viruses that get made. And as we'll see on the next figure from your textbook, sometimes those bacteriophages, instead of just packaging their own DNA, they will package some DNA from their host cell. And then that bacteriophage can leave the original cell and go off and infect a new cell. And by doing that, it can bring over some DNA from its original host. So here's a depiction of that from your textbook that you want to take a closer look at that while you're reading. So this is a bacterial cell. Here's a virus, this thing that kind of looks like an alien, and some of them really do look like that. And that alien structure is built from different kinds of proteins, so it's kind of like a little miniature micro protein spaceship. And what they're showing you here is the bacteriophage docks onto the surface of the cell and injects its DNA into this unsuspecting bacterial host. Once the DNA is in there, the DNA, that genome, has genes on it that code for the parts of this bacteriophage. So the bacteriophage's genome has to be replicated. You know, we've talked about DNA replication before. And also, the protein parts of the phages are being made inside this bacterial cell. And then we wind up assembling new bacteriophage viruses that have been produced inside that cell. And in their little heads up here, they're going to package the genome of the bacteriophage. But every once in a while, that's what they're trying to show us over here, pieces of the host cell DNA, they're showing you right there, the little piece got separated. Every once in a while, a piece of the host cell DNA will go into that bacteriophage head. Okay, and so eventually this cell that's infected with these bacteriophages will burst. That's called lysis. Those bacteriophages spill out. And then this is the one that has a piece of bacterial host DNA in it. So it finds a new cell to infect, injects that DNA from the donor cell up here, and now this recipient cell has picked up a piece of bacterial DNA from the previous host, And by doing that, it might acquire some new traits that it didn't have before. So this is another method by which some bacterial DNA from one cell ends up in a new cell that didn't have that DNA before. It's just that in this case, it's happening by way of a virus called a bacteriophage that's helping that process along. So this is called transduction. So we've talked about transformation. That's where a bacterial cell on rare occasion takes up a piece of naked DNA in its environment and acquires new traits. We talked about conjugation. Some bacteria can send out a sex pilus and transfer some DNA over to a recipient cell. And third, we've talked a little bit about transduction, where bacteriophages can mistakenly transfer a piece of DNA from one bacterial host cell into a new one. And, in all of these instances, these recipient cells may acquire new traits that they did not have before. So there are some known examples that are important clinically where transduction was involved. So a good example is Corynebacterium diphtheriae or diphtheriae, a couple of different pronunciations for that. That causes the very severe respiratory infection, diphtheria. However, there are different strains of Corynebacterium diphtheriae. Some do cause the respiratory illness and some don't. The ones that do contain genes that code for a toxin, a bacterial exotoxin that damages the respiratory tract and causes this horrible illness. And so it has been discovered that gene for that toxin can be transferred through transduction from a cell that doesn't have it, from a cell that has that gene to a cell that didn't have it before. It can be passed through a bacteriophage. There are various Clostridium bacteria that do this, where transduction has been involved in moving genes from cells that didn't have them, from cells that did have them to cells that didn't have them. And then there are strains of Streptococcus pyogenes, that's your strep throat organism. There are strains of that have picked up new genes through transduction as well. All right, here's a little Q&A for you. This is a true story. E. coli, Escherichia coli, is a bacterial organism that's naturally found in the colon, the human large intestine. And E. coli, even though we hear negative things about it sometimes in the news, it's generally a beneficial organism. So it's living in your colon, it's blocking bad types of bacteria from being able to live there, makes vitamin K for us, helps you to digest some of your fiber. So a good organism to have around. But there are strains of E. coli that are genetically a little bit different, and they have traits that turn them from a bad type of bacterial organism, turn them from being a good type of bacterial organism into a bad one. And an example of that is E. coli O157H7. That is a strain of E. coli that can cause a very severe gastrointestinal illness, severe diarrhea, very painful. And it's been discovered that E. coli O157H7 produces a protein toxin called the Shiga toxin. So regular E. coli doesn't make that toxin. It's a protein. This strain does. And it's also been discovered that E. coli acquired that gene from a cousin, Shigella. So Shigella is a genus name for some gram-negative bacteria that are very similar to E. coli, but Shigella bacteria make this nasty toxin that's capable of making people very sick. And at some point, a regular old, happy, friendly E. coli cell picked up the gene coding for the Shiga toxin from a Shigella cell in its environment through horizontal gene transfer. So this really does have clinical relevance out in the wild. And here we actually had horizontal gene transfer from one species of bacteria, Shigella, and even from one genus, from the Shigella genus to the Escherichia genus of bacteria. So sometimes it's not even between members of the same species. Sometimes these events can cross from one species to another one. Okay, so we've talked about some example horizontal gene transfer methods. That's one of the methods by which bacteria, we've really been focusing on bacteria here. This is a method by which bacteria can change genetically, and sometimes those changes are harmful, but in some instances they're beneficial for survival. In the next video lecture, we're going to talk more specifically about genetic mutations and how those can alter the traits that an organism has. With horizontal gene transfer 2 and recombination, there are some instances where that happens in other types of microorganisms as well. We're not going to be able to get into that, but for example, viruses can exchange genetic information with each other and undergo recombination events in some cases. Influenza is a great example of that, and we'll talk a little bit more about that when we get into Chapter 5. So I don't want you to think that horizontal gene transfer only occurs in bacteria, but we really have honed in on how that does occur in some types of bacteria and the relevance to pathogenic types of bacteria that can make us sick. But in the next video clip, we'll get more into mutations, and genetic mutations apply across all living things as a mechanism for genetic change.